Good afternoon. All right. I'm going to introduce everyone to Regenxbio. Thanks very much for the organizers for inviting us back to present. We're a gene therapy company at a ophthalmology conference, and we are a public company based in Rockville, Maryland. We have over 250 employees, a number of investigator partners, number of company partners working on over 30 drug development programs worldwide. We have a plan to bring products to market based on gene therapy and a proprietary technology, and we have a science that's about the delivery of genes to treat diseases using proteins that are expressed from those genes associated with genetic mutations, but also therapeutic antibodies. Since we're a public company, I may be making some forward-looking statements about the background of Regenxbio. As I mentioned, we started as a company focused on science of delivery. We've expanded the application of that delivery within the walls and outside of the walls of Regenxbio, and today, inside the company, we have 4 clinical stage programs focused on 3 separate therapeutic areas. There's one FDA-approved product based on Regenxbio technology that was approved for a neuromuscular disease back in May of this year, the first approval for a life-threatening disease of a gene therapy in the US, and we have partners developing 15 clinical stage products. The NAV technology platform has an application to go well beyond ophthalmology, and the slide that I'm going to show you about the internal development program at Regenxbio represents that. We have a lead program using NAV technology and gene therapy to treat wet age-related macular degeneration, but we're also inside the company focused on neurodegenerative diseases and liver-directed diseases that tend to be more focused on that gene mutation side of the drug development process. But as a company, because we're gene therapy, we've thought a lot about how to get products moved forward in the gene therapy space and paradigm. And I'm going to apply some of that thinking today to how we're approaching that in ophthalmology for a large disease like wet AMD. Our partners are really advancing the technology in a variety of different indications as well. And as I mentioned, that one product that's on the market today for SMA type 1, marketed by Novartis in the US, but 15 additional programs are in clinical development. This morning, I had an opportunity to meet with one of the CEOs of one of our partner companies based here in San Francisco and get some feedback and talk about the experience they were having with a rare disease. And just to give you an impression of the type of innovation and influence that gene therapy is having in drug development, we're seeing treatments that are coming into drug development with virtually no traditional target discovery or traditional drug discovery. We're seeing genes get identified and the product being the gene and then accelerating right into the clinic. And when that happens, we need to learn a lot very fast. But what can happen, especially in some of these rare diseases, is that they can accelerate because some of the clinical benefits can really be outstanding. The approval of something like the SMA product, for instance, took roughly 5 years from the first time that the first patient received a dose. And the CEO I was talking with this morning is in a similar situation, focused on a very rare and life-threatening disease in children that may see a similar type of timeline for development. So taking advantage of that innovation, in science and the innovative thinking in the regulatory space, the question is, how can we apply that to larger indications? And that's what I'm going to try to talk you through today in terms of how we're thinking about that with our technology platform here at Regenxbio. We're developing a treatment called RGX-314 for wet age-related macular degeneration. It is a gene that encodes a monoclonal antibody fragment against VEGF. And it's using our proprietary technology to be delivered to the back of the eye to use the cells in the eye to express that protein and basically provide that protein in the same way that many of you, I'm sure, in this room are applying that protein with regular anti-VEGF injections today. And Aaron provided a good insight already into sort of the paradigm of thinking here, which is one-time treatment that is durable, but that is safe and effective, like the treatments that are available today. We've chosen to take forward this clinical candidate on the basis of looking at the anatomy of the eye and looking at the physics and the biology of how to deliver something into the eye and what AAV can actually do. And I tend to come from a world before I was in gene therapy that was associated with diagnostic instrumentation and life sciences research tools. And the one thing I always learned there is that physics usually trumps biology. No offense to the biologists in the room. And in this case, when we're trying to deliver AAVs into the eye, we do feel that it's important to safely get it to the most effective spot near the course of the disease. And so for us, we took advantage of sort of known and experienced physicians, as well as surgeons in the space, to focus on drug delivery using techniques that were already well understood. In the case of our current program, we're focused on subretinal administration. Subretinal administration, many of you probably understand, is the basis of the current only approved product in gene therapy for ocular administration, which is Luxturna for the treatment of RPE65 deficiency. But recently, we've also engaged further with additional partner, Clearside, to focus on a suprachoroidal administration that we've not taken forward yet clinically with human testing, but we think has very, very similar profiles in terms of the physics and biology of getting AAV vectors and therefore the protein that expresses that anti-VEGF molecule into the right space safely and effectively. And we're looking forward to updating more on that as our programs advance. We have a clinical trial ongoing with the subretinal route of administration. And the takeaway from this clinical trial is that we're advancing into wet age-related macular degeneration with a one-time treatment, that we're looking for safety, efficacy, and a lot of information about the pharmacology of our treatment early on. We're going into very severe wet AMD. We're looking at patients with the subretinal administration. We've treated 42 patients in our trial, and as of AAO last year, we reported data on 18 of those patients. Tomorrow, we're going to update on the trial on the additional 24 patients that have been dosed since last year. And today, I'm going to just quickly take you through where we are with, again, the current 18 patients that we reported on last year. RGX-314 has been well tolerated. We have not seen any inflammation associated with the drug. We have a profile that we believe is supported across all dosing cohorts. In terms of secondary endpoints in our trial, it was important for us to measure protein to understand not just the pharmacology of how much of the delivery vehicle we were putting into patients, but also to correlate that to how much anti-VEGF protein we were producing in the eye. The only way we could think of doing that in vivo in patient subjects was to pull some fluid from the anterior, even though our drug is being delivered to the back of the eye. What we've seen is dose-dependent increases in protein from the first 6 patients in the first cohort all the way to the last 6 patients in the high-dose cohort, cohort 3 at this point in time. And with the dose-dependent increases in protein, we see reductions in the need for VEGF injections for patients that were coming into the trial previously high-need VEGF users, as well as maintenance of vision and pathology. We've shown data out to 1 year in these cohorts and talked about the fact that in our third dose cohort, we have 3 of the 6 patients that have continued in the study to receive 0 injections for 18 months and had gain in vision and maintenance of pathology. And here's the 1-year data on the BCVA and CRT for those patients. We're planning to advance RGX-314 further outside of wet AMD. The program is moving forward into a new clinical trial this year for diabetic retinopathy, still using the subretinal route of administration, which we think we've established as safe, and now feel like we can move that into additional disease indication. So I think when I reflect upon where we are with gene therapy, I reflect about where we are with respect to the RGX-314 program. Having dosed 42 patients with 5 different doses of gene therapy, measured protein in all of those patients, looked at secondary measures like vision, pathology, and change in injection need, I believe that we have one of the most robust programs for establishing and understanding the pharmacology of a drug that I've seen for wet AMD and gene therapy. And I believe that to be incredibly important because we're talking about going into a space where gene therapy has not gone before. As I mentioned when I was talking to my colleague this morning about their rare disease experience, we're talking about trials that are supporting approvals that involve 15 patients or less, single-arm studies, no controls. There's a lot of forgiveness in the rare disease space. There's a lot of innovation happening in gene therapy. But for us to be able to advance gene therapy into larger indications to help even more people with a background of a disease like wet AMD, we really need to have the level of robustness in early stage programs that hasn't existed at this point, and that I believe Regenxbio is committed to. So thanks a lot for your attention today.